Let's talk about radioimmunoassay, or also known as RIA. The radioimmunoassay was made by Yellow and Berson. In 1959, the researchers developed a system that used a radioactive label to identify low amounts of immune components. Because of inherent drawbacks of using radioactive isotopes as labels, the researchers began to search for a substitute for the successful RIA method in 1960s. Chemiluminescence technology has sensitivity at least as good as that of an RIA, which is relatively simple and cost-effective. An, an older and less commonly used method is RIA, or radioimmunoassay. To develop immunoglobulins, RIA uses radioactive substances. Specific antibodies in liquid solution are used in traditional RIA. Traditionally, RIA is performed using particular antibodies in liquid solution. Solid phase RIA makes use of antibodies that have been coupled to a solid support. Radioimmunoassay technique is a very sensitive in vitro technique used to measure the concentration of antigens through the use of antibodies directed against these antigens. In performing radioimmunoassay, a known amount of an antigen is labeled with the gamma radioactive iodine isotopes such as 125I, linked to tyrosine to make it radioactive. This radiolabeled antigen is then combined with a specified amount of antigen-specific antibody, and two selectively attached to one another. Then a patient's serum sample having an unknown amount of that same antigen is introduced. As a result, the unlabeled or cold antigen from the serum competes for antibody binding sites with the radiolabeled antibody or radiolabeled antigen or hot. As the quantity of cold antigen increases, more of it binds to the antibody, displacing the radiolabeled variant and lowering the antibody-bound radiolabeled antigen or free radiolabeled antigen ratio. The bound antigens are then separated and the radioactivity of the remaining free unbound antigen in the supernatant is quantified with a gamma counter. In principle, this technique may be applied to any biological molecule and is not limited to serum antigens. It is also not needed to employ the indirect way of measuring the free antigen rather than directly measuring the trapped antigen. For example, if radiolabeling the antigen or target molecule of interest is undesirable or impossible, an RIA can be performed if two distinct antibodies that identify the target are available and the target is big enough to present several epitopes to the antibodies. One antibody would be radiolabeled in the manner described above, while the other would be left unaltered. The RIA would begin by allowing the cold unlabeled antibody to interact with and attach to the target molecule in solution. This unlabeled antibody should be immobilized in some form, such as being linked to an agarose bead, coated to a surface, and so on. The initial antibody-target molecule combination is then permitted to interact with the hot radiolabeled antibody. Following thorough washing, the direct quantity of radioactive antibody bound is measured, and the amount of target molecule quantified by comparing it to a reference amount also measured at the same time. In theory, this approach is identical to the non-radioactive sandwich ELISA or ELISA method. Enzyme immunoassay or EIA uses a non-isotopic label and is safer but shares the specificity, sensitivity, and rapidity of RIA. This is a diagram of an RIA. Step 1, it adds buffers to the tubes. Step 2, add known amounts of unlabeled antigen to the mixture. This compete for the binding sites of the antibodies. Step 3, add radioactive antigen to the mixture. Step 4, add fixed amount of antibody to the tubes. Step 5, radioactive antigen is displaced from the antibody molecules by the unlabeled antigen. Precipitate AgAb complexes with PEG secondary antibody. Lastly, the antibody-bound antigen is separated from the free antigen in the supernatant fluid and radioactivity of each is measured. Radioimmunoassay detects the following: narcotic drugs, hydromorphone and hydrocodone, flunisolide, ferritin, digoxin, and thyroid testing. Its are it is specific, it is sensitive, convenient, reliable, and reproducible. It also has disadvantages. It is prolonged reaction time. It is costly because it uses radioisotopes. It also has health hazards due to radioisotopes, limited assay range, lack of direct linear relationship, difficulty in automation, and it has lengthy counting time. That is all for radioimmunoassay.
Chemiluminescence. Chemiluminescence refers to light emission produced during a chemical reaction. It is used extensively in automated immunoassays. This methodology has excellent sensitivity and dynamic range. It does not require sample radiation, and non-selective excitation and source instability are eliminated. In chemiluminescence, most reagents and conjugates are stable and relatively non-toxic. In immunoassays, chemiluminescent labels can be attached to an antigen or antibody. Acrylium esters are highly specific activity labels that can be used to label antibodies and haptens. Also, chemiluminescent labels are used to detect proteins, viruses, oligonucleotides, and genomic nucleic acid sequence in immunoassay. In chemiluminescence, there are two formats used. First, competitive immunoassay. It has a fixed amount of labeled antigen competes with unlabeled antigen from a patient specimen for a limited number of antibody binding sites. The amount of light emitted is inversely proportional to the amount of analyte or antigen measured, which is seen in this photo. Second, sandwich immunoassay. The sample antigen binds to an antibody fixed onto a solid phase. A second antibody, labeled with a chemiluminescent label, binds to the antigen-antibody complex on the solid phase. In the sandwich assay, the emitted light is directly proportional to the analyte concentration. The detection device for analysis is a simple photomultiplier tube used to detect the emitted light, which is seen in this photo. Chemiluminescent labels can be divided into 5 major groups, which are first, luminol, second, acrylium esters, third, peroxyoxalates, fourth, dioxetanes, 5, trirudenium. Direct labels include luminol, acrylium ester, and electrogenerated luminescent chelate from rudenium and tripoxyliminene. TPA complex labels are attached directly to antigens, antibodies, or deoxyribonucleic acid probes depending on the assay format. Oxidation of isoluminol by hydrogen peroxide H2O2 in the presence of catalyst, example, microperoxidase, produces a relatively long-lived emission at 425 wavelength. Oxidation of acrylium ester by alkaline H2O2 in the presence of detergent produces a rapid flash of light lasting from 1 to 5 seconds at 525 nm. Peak intensity can be used for the measurement. An alternate method is to use an integrator to measure the entire light output for great sensitivity. Enzymes are typically used for indirect labels. Indirect labels are attached to antibodies, antigens, and DNA probes depending on the assay's format. Enzyme labels often used in indirect. Procedures include the following: alkaline phosphate, horseradish peroxidase, beta-galactidase. It is an interesting label is native or recombinant apicorine from bioluminescent jellyfish. It is activated by reaction with coalaterrazine. Light emission at 469 nm is triggered by reaction with calcium chloride. Specific clinical application. One of many clinical applications of chemiluminescent is a third-generation serum IgE method. Imulite 2000, a solid-phase bead two-step chemiluminescent EIA allergens are covalently lined to a soluble polymer ligand matrix, allowing immunochemical reaction to occur in a liquid phase for random access automation. Using the RUBPY3 plus complex label, various assays have been developed in a flow cell using magnetic beads as a solid phase. Beads are captured at the electrode surface and unbound label is washed out of the cell by wash buffer. Label bound to the bead undergoes an electrochemiluminescence reaction, and the emitted light is measured by an adjacent photomultiplier tube.
Hello everyone, I am IS Christine Torio from Bachelor of Science in Medical Laboratory Science, a third year student. So today, we will discuss about immunofluorescence. So first, what is immunofluorescence? The fluorescent labeling is another method used to demonstrate the complexing of antigens and antibodies. It is a method in biology that relies on the use of antibodies chemically labeled with fluorescent dyes to visualize molecules under a light microscope. Fluorescent molecules are used as substitute for radioisotope or enzyme labels. The fluorescent antibody technique consists of labeling antibody with fluorescein isothiocyanate, a fluorescent compound with an affinity for proteins to form a complex. This conjugate is able to react with antibody-specific antigen, and the antibodies may be conjugated to other markers in addition to fluorescent dyes. The use of these markers is called colorimetric immunologic probe detection. In addition, these reagents have the advantage of requiring only a standard light microscope. Let us now move on to the three basic methods in fluorescent conjugates. First off, the direct immunofluorescent assay. This is a technique used in the laboratory to diagnose diseases of the skin, kidney, and other organ systems. Take note that it is a conjugated antibody that is used to detect antigen-antibody reaction at a microscopic level. The DFA can be applied to tissue sections or in smears for microorganisms. Fluorescein conjugated antibodies bound to the fluorochrome FITC that are used to visualize many bacteria in direct specimens. HRP conjugated to antibody, the immunoperoxidase stain, can be used to detect CMV, or the other viruses or D-nucleic acid in cells. In biotin avidin, enzyme conjugated methods are the single-stranded nucleic acid probes, antimicrobial antibodies, or antibiotin antibodies that can be bound to the small biotin molecule. These molecules have the strong affinity for the protein avidin, which has 4 binding sites. Next is the inhibition immunofluorescent assay. The inhibition immunofluorescent assay is a blocking test in which an antigen is first exposed to an unlabeled antibody, then to a fluorescent antibody, and is finally washed and examined. Take note that if the unlabeled and labeled antibodies are both homologous to the antigen, there should be no fluorescence. This result confirms the specificity of the FAA technique. An antibody in an unknown serum can also be detected and identified by the inhibition test. And lastly, the indirect immunofluorescent assay. The indirect immunofluorescence assay, or also called secondary immunofluorescence, this is a technique used in laboratories to detect circulating autoantibodies in patient serum. It is used to diagnose autoimmune blistering diseases. Take note that the basis for indirect fluorescent assay, or the IFA, is that antibodies, or the immunoglobulins, not only react with homologous antigens but also can act as antigens and react with anti-immunoglobulins. The IFA is the serologic method most widely used for the detection of diverse antibodies, and the immunofluorescence is used extensively in the detection of autoantibodies and antibodies to tissue and cellular antigens. And some additional information: immunofluorescence can also be used to identify specific antigens on live cells in suspension. When live stem cell suspension is put through a fluorescence activated cell sorter, or the FACS, which measures its fluorescence intensity, the cells are separated according to their particular fluorescent brightness. This technique permits the isolation of different cell populations with different surface antigens. Second, the IFA, or the immunofluorescence assay, or the indirect immunofluorescence assay. The antigen to the specific antibody being tested is affixed to the surface of a microscope slide. The patient's serum is diluted and placed on the slide to cover the antigen source. If antibody is present in the serum, it will bind to its specific antigen. And lastly, the chemical manipulation in labeling antibodies with fluorescent dyes to permit detection by direct microscopic examination does not seriously impair antibody activity. The ability of fluorescent antibody conjugate to react specifically with its homologous antigen. That's all for immunofluorescence assay. Thank you for listening.
I'll be talking about regimmunoassay. The regimmunoassay was made by Yellow and Berson. In 1959, the researchers developed a system that used the radioactive label to identify low amounts of immune component. Because of the inherent drawbacks of using radioactive isotopes as labels, researchers began to search for a substitute for the successful RIA method in 1960s. Chemiluminescence technology has sensitivity at least as good as that of an RIA, which is relatively simple and cost-effective. To evaluate immunoglobulins, RIA uses radioactive substances. Specific antibodies in liquid solution are used in traditional RIA. RIA is traditionally performed using particular antibodies in liquid solution. Solid phase RIA makes use of antibodies that have been coupled to a solid support. Regimmunoassay technique is a very sensitive in vitro technique used to measure the concentration of antigens through the use of antibodies directed against these antigens. So how is regimmunoassay performed? First, a known amount of an antigen is labeled with gamma radioactive iodine isotopes. such as 125I, linked to tyrosine to make it radioactive. This radiolabeled antigen is then combined with the specified amount of antigen-specific antibody, and the two selectively attach to one another. Then a patient's serum sample having an unknown amount of that same antigen is introduced. As a result, the unlabeled or cold antigen from the serum competes for antibody binding sites with the radiolabeled antigen or HAT. As the quantity of cold antigen increases, more of it binds to the antibody, displacing the radiolabeled variant and lowering the antibody-bound radiolabeled antigen or free radiolabeled antigen ratio. The bound antigens are then separated, and the radioactivity of the remaining free unbound antigen in the supernatant is quantified with a gamma counter. In principle, this technique may be applied to any biological molecule and is not limited to serum antigens. It is also not needed to employ the indirect way of measuring the free antigen rather than directly measuring the trapped antigen. For example, if radiolabeling the antigen or target molecule of interest is undesirable or impossible, an RIA can be performed if two distinct antibodies that identify the target are available and the target is big enough to present several epitopes to the antibodies. One antibody would be radiolabeled in the manner described above, while the other would be left unaltered. The RIA would begin by allowing the cold unlabeled antibody to interact with and attach to the target molecule in solution. This unlabeled antibody should be immobilized in some form, such as being linked to a agarose bead, coated to a surface, and so on. The initial antibody-target molecule combination is then permitted to interact with the hot radiolabeled antibody. Following thorough washing, the direct quantity of radioactive antibody bound is measured, and the amount of target molecule quantified by comparing it to a reference amount also measured at the same time. In theory, this approach is identical to the non-radioactive sandwich or ELISA or ELISA method. Enzyme immunoassay or EIA uses a non-isotopic label and is safe, but shares the specificity, sensitivity, and rapidity of RIA. So as I said, this diagram, it adds buffers to the tubes in step 1. Step 2, add known amounts of unlabeled antigen to the mixture. These compete for the binding sites of the antibodies. Step 3, add radioactive antigen to the mixture. Step 4, add fixed amount of antibody to the tubes. Step 5, radioactive antigen is displaced from the antibody molecules by the unlabeled antigen. Precipitate AG, AB complexes with PEG secondary antibody. Step 6, the antibody-bound antigen is separated from the free antigen in the supernatant fluid and the radioactivity of each is measured. So that's how radioimmunoassay is performed. So what does RIA detect? It detects narcotic drugs, hydromorphone and hydrocodone, flenisolide, ferritin, digoxin, and on thyroid testing. What are its advantages? It is specific, sensitive, convenient, reliable, and reproducible. It has its disadvantages. It has prolonged reaction time. It is costly because radioisotopes are used. And because of radioisotopes, it has health hazards, limited assay range, lack of direct linear relationship, difficulty in automation, and it has lengthy counting time. That is all for radioimmunoassay.